Now, let's summarize this functional unit of the kidney, nephron. So first, afferent arteriole gets into this glomerulus and it exchanges as a filtration, and then it gets out of efferent arteriole of blood. And those filtered part will be coming out of this and in the Bowman's space. So from inside point of view, looking at the capillary, very interestingly, this capillary has lots of holes. And look at this, this is 200 nanometers, so this is about 100 or less. So it's about 90 nanometer pore. That's in kidneys, endothelium itself formed as a physical filter, which is very amazing to see. And in the end, so this happens in this filtration, but later on the kidney are filtered, which will become a urine. But from there, as you saw, that water and vital ions will be reclaimed, called reabsorption into the blood. And those who, what we don't need it, will be secreted into the, combined into the urine. And these are renal artery part and the renal vein part. And people have figured out each kidney, we have two kidneys, one has about 500,000 nephrons. So both of these two kidneys, we have about 1 million nephrons. So the glomerulus, and this part of is where the filtration function is happening in the kidney. And the Bowman, and so these are glomerulus, and the Bowman space is filtered plasma, which contains small molecules because it has been filtered through the glomerular membrane. And afferent and efferent arterioles are what they do is bring blood into and away from the glomerulus of each nephron. And we saw glomerular capillary bed is filtering with fenestrated endothelial cell, meaning that it's a very leaky, it has a holes in it of the capillary wall. And also for the cell, special cell type from the outside of the capillary is called podocytes. They perform ultrafiltration. So now let's see, look at a little bit of data for this molecular filtration. So there are these substances, for example, water, very important, sodium and glucose and insulin, myoglobin, and albumin. So let's look at the substance. Especially the size matters goes together with molecular weight in general. So we look at the molecular weight, and another aspect is how well these become filtered through this glomerulus. So if it's just a freely filtered, coming back and forth, because it's so small, its filterability is 1. So what matters is first size, purely size. Sodium has molecular weight of 23, that's perfectly filtered, which means in and out, the concentration through this glomerulus is the same. And what about glucose, which is very important for us, and it's also filtered out, okay? And insulin has about 5.5 kilodalton, and it's also still too small, its filterability is 1. And myoglobin is a hemoglobin-like element, especially for our muscle, to keep the oxygen to deliver and keep and deliver oxygen, which is about 17 kilodalton. Now it gets bigger, and the filterability is not 100%, about 75%. And let's consider albumin, which is about 69 kilodalton, which is pretty big, and that corresponds to a molecular radius of about 3 nanometers. Still tiny compared to that 90 nanometer hole of the filters in the kidney. But look at that, the filterability is almost 0.005, less than 1%. What happens here? So let's think. So the first filtration is size. So water and small molecules pass through the pores in the glomerular membrane. And large molecules cannot pass through the pores. So that we can because it's large, you can't pass through the filter. So we can define a measure of fraction of these molecules that would pass through the membrane called filterability. And what's also people have figured out is, charges can matter for this, not only the size. So people have studied this, and here's the data looking at effective molecular radius in x-axis, y-axis is a clearance ratio, meaning that if it's 1, it'll be totally cleared out. So the dextran, representative molecule of dextran, neutral dextrans, you see, the bigger, the less it's cleared out. Okay. What's interesting here is cationic dextran versus anionic dextran. So if the dextran has a positive charge, it's somehow cleared easier compared to the negatively charged. I do not know why, but I guess the cell membrane usually are having negative charge, and that may be why. So charge, as you here see, negatively charged molecules are more hindered, more difficult to filter. So a good example is albumin. This protein is almost less than 1% is filtered. So which is good, that albumin keeps inside the blood. And in fact, what happens is in the blood, albumin is one of the most abundant protein, not one of the, it's the most abundant protein in our blood plasma. And the remaining albumin is in fact doing very important role for reabsorption, because in the proximal tuber and distal tuber, those filtrates, because of the osmotic pressure and generated, the water gets back to the blood because of the osmotic pressure gradient. So, for example, human serum albumin is about 50% of human plasma proteins, and its function is to maintain osmotic pressure of blood so that it can reabsorb. Because otherwise, with this filterability, water, almost 100 liters will come out through the ureter, and it's not happening because of this albumin. Okay.